Welcome to The Explainer. Today, we're diving into the incredible science of how we make the invisible, well, visible. We're talking about the world of bacteria. And it all kicks off with one really basic but super important question. So think about it. This is the big challenge for microbiologists everywhere, right? They're trying to study these tiny organisms, things you can't even see at all, but things that can have a huge, huge impact on our health. So, yeah, how do you even begin to study an enemy you literally can't see? Well, the solution is actually pretty clever, and honestly, kind of simple. You can't see just one little bacterium, right? But what about a million of them? Or a billion? That you can see. And that's the whole game right there. The science is all about giving them a place to grow and multiply until they show up as these visible little communities we call colonies. So to build that perfect little microbe paradise, scientists basically become chefs. They've got this whole cookbook of special recipes, and each one is designed to grow a very specific kind of microscopic life. And that recipe, well, its official name is culture media. And you can really think of it just like that, a specially prepared food. It's this nutrient-rich stuff that scientists mix up in the lab to give those microbes everything they could possibly want to eat to grow and just multiply like crazy. And you know, the whole process is pretty straightforward. It really breaks down into these 4 key steps. You get your sample, say from a throat swab. Then you inoculate the media. And that's just a sciencey word for spreading the sample onto that special food we just talked about. After that, you incubate it, which just means you stick it in a nice warm place so the microbes can grow. And finally, you analyze what pops up. Okay, so this microfood basically comes in 2 main styles. First, you've got your liquid media or broth. Think of it like a nutrient soup. It's awesome for when you just want to grow a massive batch of microbes all at once. Then there's solid media. This is more like a Jell-O-style surface. And what's cool about this is that it lets individual microbes grow into their own little separate colonies, which we can then see, count, and tell apart. Now, the secret ingredient that makes this whole solid media thing work is something called agar. It's a gel that comes from seaweed. But here's the really interesting part. The agar itself isn't the food at all. It has 0 nutritional value for the microbes. Its only job is to be the plate. It just provides that solid surface, turning the nutrient soup into a kind of microbial buffet. But of course, it's not that simple. You can't just have one recipe for everything. See, microbes are a lot like us. Some of them will eat anything, but others are incredibly picky eaters. So to deal with that, scientists have come up with a whole toolkit of specialized media, each one for a different purpose. Okay, first up in the toolkit, we have what's called enriched media. I like to think of this as the all-you-can-eat 5-star buffet. You start with the basic nutrient mix, but then you add extra, fancy stuff like blood or serum. This is for those fastidious microbes, which is just the scientific term for the really, really picky ones that refuse to grow without the gourmet treatment. Next, you've got selective media. This one is like the bouncer at a super exclusive nightclub. It's packed with special ingredients, things like antibiotics or salts that literally block other unwanted bacteria from growing. It creates this VIP-only environment, so only the specific microbe a scientist is actually trying to find can get in and grow. And rounding out our toolkit is differential media. This stuff is so cool. It's basically a detective kit. It has these built-in chemical indicators, and what they do is cause different bacteria to show up in different colors, or to change the look of the plate around them. It lets a scientist tell different microbes apart just by looking at them, even when they're growing right next to each other. Okay, so these different types of media are cool in theory, but how does this all play out in the real world? Let's take a look at a classic case study. Something that's used in medical labs every single day. So check this out. We're looking at blood agar and chocolate agar. And at first glance, they seem almost identical. Both are made with nutrient agar and sheep's blood. But here's the kicker. The only difference is the temperature during preparation. For blood agar, the blood is added when the mix is cool, around 45 Celsius. For chocolate agar, they add the blood when it's much hotter, about 70 degrees. And that extra heat actually cooks the blood cells, causing them to break open and release even more nutrients. It also turns the plate brown, which, you guessed it, is why it's called chocolate agar.
And this brings up a really important point. A single type of media can actually do more than one job. It can wear two hats, so to speak. Blood agar is the perfect example. On one hand, it's enriched, right? It's got that blood to feed the picky bacteria, but it's also differential because it lets us see how the bacteria interact with the blood, which gives us a ton of information.
So how does it do that? Well, it's through a process called hemolysis. It sounds complicated, but it's not. Hemo means blood, and lysis means to break apart. So hemolysis is just the breakdown of red blood cells. Some bacteria release these toxins that actually destroy the red blood cells in the agar right around their colonies, and that creates these really obvious visual patterns.
And these patterns are like fingerprints. They're major clues for identifying the bacteria. You have beta hemolysis, where there's a complete clear zone around the bacteria, total destruction. Then there's alpha hemolysis, which is a partial breakdown that looks kind of greenish. And finally, gamma hemolysis, which means nothing happened. No breakdown at all. Just by seeing which of these patterns shows up, a lab tech can get a huge head start on figuring out what they're dealing with. Okay, so this is all really fascinating lab stuff. But let's bring it back to you. Because this whole process, from cooking up the media to reading those patterns, isn't just a trick. It is a fundamental part of modern medicine that affects all of us. I mean, the impact is massive. We're talking about diagnosing things like strep throat, figuring out exactly which antibiotic is going to knock out your infection, isolating bacteria to develop new vaccines. Even just getting a delicate sample from the doctor's office to the lab safely relies on these techniques. It's everywhere. So here's the bottom line. The next time you have to get a throat swab, you'll know exactly what's happening. That simple swab is the first step. It's going onto one of these special recipes, onto a culture media, so that the invisible world causing the problem can be made visible. It's the bridge between your symptom and a clear diagnosis and the right medicine. It's how we see, understand, and fight back. And that really leaves us with one final big question to think about. This is a constant race, right? An arms race between us and the microbes. They're always evolving, always getting smarter, developing resistance. So as they get more complex, what's next? What kind of new ingenious recipes will scientists have to invent just to keep up?